So, our chief scientist, Dr. Jack Kessler, who really is arguably the most renowned iodine chemist on the planet, has been formulating iodine chemistries for 35 plus years. And this technology is the culmination, if you will, of his life's work, which has brought about a platform that is far above anything that has been done before. Well, simply what we've done is we have isolated and stabilized molecular iodine. And it's important to understand that in iodine chemistry, in different iodine formulations, the only active molecule in that formulation is molecular iodine. It's the only thing that kills pathogens. And in the traditional iodine chemistry, greater than 99.9% of the iodine species are there to just stabilize the active because it's so unstable in formulation. And molecular iodine gets absorbed into human skin and then it gets off gassed. And of course, the molecular iodine is going to kill pathogens on contact, but we can turn the skin safely into a biocidal material for up to 24 hours or more. And it's this off gassing of the molecular iodine that is creating this persistent barrier to pathogens. So when we talk about things like skin prep for surgeries, we can help diminish greatly the number of surgical site infections. When we talk about diseases of the skin, not only are we putting the product on to treat at that moment, but because that is being off-gassed, it is persistently killing the pathogens which are creating that disease state. And in pilot studies and in anecdotal evidence, we've seen rapid healing of topical diseases. And what is extremely exciting to us is that we have created IP that allows us to not only have the skin absorb molecular iodine, but we're able to partition molecular iodine into polymers. So imagine a catheter that is biocidal for up to a month. And when you think about all of the types of infections that are involved with urinary catheters, peripheral catheters like IVs, central lines like PICs, there's a whole host of polymers that are used to get into the human body's circulatory system, digestive system, things like that. But we knew because we were going to load lots of molecular iodine into a formulation or into a polymer that we weren't going to have a problem being effective. But we've also been shown three characteristics that we weren't expecting. When the product has been used on wounds, wounds have been healing dramatically faster than previously seen before. So it's having a wound healing property. When those wounds heal, there's a large reduction or or actual absence of scarring. And then thirdly, it's been acting in many instances like a very powerful anti-inflammatory. So much so that the anti-inflammatory and wound healing nature is part of the IP that has been written into our second patent application. And we know that growing a company organically would, one, it would require a lot more funds to do. And then also creating a sales environment, creating a sales force, a marketing organization, it would just extend the commercialization of each and every one of these products. And we are looking for not the generally the number one or the number two market leaders, because generally they have very large revenues that they need to protect. We're looking for number 3 through 7 that are looking to capture market. And when they find a innovative, very disruptive technology, they are willing to bring that across the finish line because they want to capture market share. And our business model allows us to license um, our technology where we are getting a license fee, where we are always going to manufacture the molecular iodine. The IP will always remain with I2Pure. So when we sell the molecular iodine to a partner, they are blending and bottling very much like a Coca-Cola would deliver their syrup to a bottling company, which would then just add carbonated water to that mixture and then bottle it. And then we would receive a royalty as well. And then in some instances, like with the wound irrigation system, I2Pure is participating in that LLC that's creating um, a entity that we will be again selling the molecular iodine to, receiving a royalty from, but as that company grows and a potential exit, we will share in the profits of that company as well as the potential exit from that company. So we can play on both sides of that equation. So there are two pivotal studies that have collected all of this information, and just referencing the peripheral catheters, the IVs that people get. There are 2 billion of those placed every year worldwide. Out of those 2 billion, 4.33% are creating catheter related bloodstream infections. Of those infections, 9.4% of those people are dying from directly related to that bloodstream infection caused by the catheter, which is equivalent to 8.1 million deaths a year from IVs. Well, the catheter market globally is a $51.6 billion market. And there are things that clinical organizations are trying to do to prophylactically prevent lots of infection, and they're just not effective. So when you think about a $51.6 billion market and a catheter that could potentially prevent these infections, it's quite dramatic. About 10 days ago, we received our first third-party verified study um, from a world-renowned laboratory, and we sent them catheters which they tested for biocidal efficacy. The two strains of bacteria were E. coli and staph, and we sent them the catheters. They took the catheters, they rinsed the catheters under tap water until the catheters ran clear. They dried them until there was no discoloration on the paper towels. And then they placed them in the material that was inoculated with both those strains of bacteria. And within that test parameter, which has a sensitivity of up to greater than 4 log reduction or greater than 99.99% effective, we maxed out the test in both with both strains of bacteria. So we know the catheter is going to do a remarkable job killing pathogens. Um, we know from prior third-party experiments when we were talking about a liquid material that is roughly 300 times more powerful than the current skin prep that they're using in the OR, that we are also non-toxic to human cells in a 24-hour human in vitro cytotoxicity study. So not only are we incredibly effective, we're also incredibly safe. The traction in the catheter market is that we are speaking to three publicly traded multi-billion dollar companies that have started to kick the tires. We are talking to some of the largest healthcare delivery systems in the US, trying to find the appropriate healthcare system to co-develop the final product. It has attracted lots of attention. So wound care, and there's lots of different chronic wounds, but approximately it's a $28 billion market, and it includes things like diabetic foot ulcer and pressure ulcers and infections from healthcare activity of wounds that just don't heal. And the problem is that up to 85% of these chronic wounds have biofilms in them. And just without getting into lots of science, just consider biofilms very, very hard to kill substances. It's a pathogen, it's a bacteria or a fungus that creates a castle for itself, an armored vehicle, if you will. And biofilms with the pathogens inside, they stop using oxygen, they stop replicating, and they build this fortress. And then once the fortress is built, they start sending out these planktonic attacks of pathogens. And they're very, very difficult to kill. So when you're healing a wound and you have biofilm involvement, you have to do two things. You have to heal the wound, so the tissue needs to be healthy, and then you have to kill the biofilm, which does not go hand in hand with keeping tissue healthy, because most of the things that destroy biofilms destroy human tissue. So we have two executed contracts, um, one in Brazil, one in the Philippines. One is $9 million over a 5 year period, and one is $36 million over a 5 year period. And we are negotiating of active contracts, probably $150 million more over that 5 year time period. And when you start talking about the conversations I'm having about the catheters, I'm in active conversations about the sutures. I'm in active conversations about several different products. And our pipeline is pretty dramatic when you think that there are roughly 700 billion plus dollars of different market segments that we could easily go into. Well, the two journals that have published medical manuscripts around our technology is the International Journal of Pharmaceutics and the Journal of Hospital Infections. And both the International Journal of Pharmaceutics, which was led by Dr. Victor Nizet out of UC San Diego, really captured the difference between molecular iodine and existing iodine formulations, demonstrated its efficacy, and then actually used mice studies, animal models, with acute dosage of their nasal passages to show that there was no irritation. So it showed that very effective and non-irritating. And then in the Journal of Hospital Infections, which was done by our chief scientist and then peer reviewed by 5 or 6 different MDs and research PhDs, we demonstrated that molecular iodine is not the toxic component of iodine formulations. So the studies that are going on right now are really lending themselves not so much in a peer review environment where it's going to be published in a journal, but they are lending themselves directly to either 510(k) applications or phase 3 trials. So we're working with major institutions to bring a dermal regeneration pad, a pad that is used for full thickness wounds that are caused by burns or traumatic injury, where the pad sits on the fascia and allows the dermis to grow up into it. That particular institution is currently finalizing this week, by tomorrow, the final studies, which will then allow us to go directly into animal studies within the next 10 days, which really is the long pole in the tent, because we have to collect a 6-month study before we can put the final 510(k) submission into the FDA. So we're hoping that submission happens within the next 7 months, and then within the next 3 to 6 months after that, the regulatory approval would be granted. We're working with the same type of procedure with a wound irrigation system, and then we are working with a top institution in North America to start Canadian phase 3 trials in May for a post-surgical nasal lavage. We are working with other institutions around the world that we have collaborations with, which are creating hydrogels, emogels, which are doing biofilm studies. So we're working, those organizations are potentially creating information that would then make it into a peer-reviewed journal. But we are really working with high-end institutions to commercialize these products to get them out as fast as possible. So, we've been truly blessed that the clinicians who have discovered us, who have started working with us, believe so strongly that we're going to have such strong patient impact that all of these studies, which I've been referencing, we have not paid a penny for, as you said. So that has been a real important aspect to our company.